see a diagrammatic representation of the steps that we have studied so far. Suppose this is a cell from which the DNA has been isolated. Let us assume that it is an animal cell. So from this cell, by suitable treatment, the DNA has been isolated. DNA has been isolated. This DNA is now subjected, let us say, to the action of a restriction endonuclease EcoR1. So EcoR1 cuts this DNA into several fragments. Wherever it identifies the palindrome sequence. Out of this, let us say this is the desired gene. That is the desired gene. Now, in the meantime, here is a bacterial cell. Here is a bacterial cell. The bacterial cell contains its own DNA, the bacterial chromosome. Apart from this, the bacterial cell has many circular DNA molecules. These circular DNA molecules are called plasmids. A suitable plasmid is identified and isolated. Plasmid is isolated. This plasmid is subjected to the action of the same restriction endonuclease EcoR1. So EcoR1 identifies the same palindrome sequence here, and therefore let us say it cuts the plasmid. It cuts the plasmid at that palindrome sequence, creating sticky ends. So a space has been created here with sticky ends. Desired gene has been here identified. It also has the same sticky ends. So now in the next step, you have to integrate the desired gene with the plasmid. Integrating the desired gene with plasmid. So the next step is integrating the desired gene with the plasmid. This is done by using the enzyme DNA ligase. So DNA ligase joins the sticky ends of the desired gene with the sticky ends of the plasmid or vehicle DNA. So this is the vector DNA. Desired gene has been integrated with vector DNA, and now you have got the recombinant DNA. That is the recombinant DNA. So the DNA that we obtained by this process is what is called the recombinant DNA. This recombinant DNA is now introduced into the host cell. Let us say E. coli, Escherichia coli. The host cell is made competent by treating with calcium chloride. So in the next step, suppose this is E. coli bacteria. This is E. coli bacteria. That is the bacterial chromosome. These are the plasmids of E. coli. Now this recombinant DNA has been introduced into E. coli. The vector that you have chosen, let us assume that it contains ampicillin resistant gene. So that is the selectable marker. So when this E. coli is allowed to undergo replication. So in the agar medium, bacterial cells divide approximately once in every 20 seconds. So in the agar medium, at the end of 20 seconds, you get 2 E. coli cells. In these E. coli cells, the bacterial chromosome would have replicated, plasmids of E. coli would have replicated, as also the recombinant DNA also would have replicated. So again, as and when this bacterial cell divides, you get a large number of cells. In each of these cells, in each of these bacterial cells, the recombinant DNA copy would be present. So you are getting multiple copies of the gene, and this is therefore also called gene cloning. So recombinant DNA is introduced into the host cell. The cell multiplies, making copies of the recombinant DNA. Then, allowing this recombinant DNA to express in the host cell. So by providing suitable conditions, the recombinant DNA is allowed to express, make its own protein in the host cell. This is done using what are known as bioreactors, vessels or tanks in which culturing of the bacteria is done on a large scale, constantly agitated, resulting in the formation of or the expression of the recombinant DNA. And finally, the product is extracted. It is extracted, it is purified, it is subjected to quality control, and maybe even preservatives are added so that you get the final product. It may be an enzyme, it may be a chemical substance, whatever it is. So all these processes are collectively known as downstream processes. So this is ultimately the technique used in recombinant DNA technology.